All right. Hi, everybody. Welcome to today's program, Less is More, the inclusion of small game in ancient human diets, featuring archaeologist and biological anthropologist and Smithsonian postdoctoral fellow, Elizabeth Grace Veatch, part of our ongoing Hot Topic series. My name is Brianna Pobener, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman wearing a black cardigan over a red shirt in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us or you've attended before, we're so glad to have you here. I want to note that we had to reschedule this program with Dr. Veatch. It was originally scheduled in March 2023, so we're very excited to be doing this today. Before we get started, a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of the Zoom interface. We're in a webinar format, so we can't see or hear you. As you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen. It has 2 speech bubbles, and then we can sort through as many as possible because the Q&A time really flies by. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with an opening presentation by our speaker, Dr. Elizabeth Grace Veatch, and then I'll join her here to take your questions. During the presentation, I'll also write answers to some of your questions, at least any that I can answer, as will our other behind-the-scenes support person, Dr. Ryan McCrae from the Smithsonian's Human Origins Program. So now I'd like to go ahead and introduce our speaker. Dr. Elizabeth Grace Veatch received her PhD in anthropology from Emory University in 2021, and she's currently a postdoctoral fellow with us in the Human Origins Program of the Smithsonian's National Museum of Natural History. Grace's interests are in early human dietary behavior, paleoecology, and foraging theory. Her primary research takes place at Liang Bua in Flores, Indonesia, and centers on understanding the foraging behaviors and diets of Homo floresiensis, and modern humans during the Pleistocene and Holocene, using taphonomic, zooarchaeological, and ethnoarchaeological methods. All right, so I'm excited to hear from Dr. Veatch.
Right. Thank you so much, Brianna, for that wonderful introduction. And I like to say I just want to start off by thanking the Human Origins Hot Topic program for hosting these wonderful videos and segments and allowing me to talk today about small animals and ancient human diets. So I'll first give some background into the topic and then show you some of my own research from Indonesia involving hobbits and giant rats. So when I talk about ancient human diets, many people may think of or envision in their head about our human ancestors scavenging meat from large animals like antelope on the African savanna. And in fact, a lot of our understanding of ancient diets comes from looking at the surfaces of animal bones for evidence of butchery. And so why are people like myself, as well as Brianna Pobener here, interested in knowing what our ancestors ate in the past? Well, one of the most visible and important trends in our lineage is the increase in brain size over time. It's actually very clear from fossil evidence that both the size and the shape of our ancestors' brains changed from something like an Australopithecus shown on the left to a modern human, which is shown on the far right. And one explanation that was proposed back in 1995 was the expensive tissue hypothesis, which basically argued that because our brains require a lot of energy, our ancestors must have changed their diet by acquiring fatty and calorically dense nutrients from larger animals. And so this hypothesis basically stated that in order to grow a large brain, the body needed to reallocate its energy from elsewhere. And so what we did was we actually reduced the size of our gut, which made it possible to energetically grow a larger brain. And so as our guts got smaller, our brains got bigger, and we simultaneously see an increase in large animal butchery from where humans were accessing bone marrow and other fatty nutrients from larger animals. And so here's just a chart to contextualize, where in time we're talking about and where our human ancestors and relatives lived, going back to around 4 million years ago. And to also give you context, modern humans are the top right red bar over on the far right. And on the bottom, you'll see a yellow bar that gives or that shows when stone tools were first used and when different stone knapping technologies, or how humans actually learned to knap and make stone tools, were invented. But what is interesting is that sometime between around 3 and 2 million years ago, we do start to see this evidence of animal bone that was being butchered using stone tools, similar to the one that's shown on your screen. And so now, whether our ancestors hunted or scavenged these large animals, which is very much debated, the prevailing narrative is that accessing these foods influenced the evolution of our culture, our technology, and our physiology, and it had a huge impact in the origins of our genus. And so one of the oldest evidence for this behavior comes from a site called Gona in Ethiopia, where several fragments of leg bones from antelope, as well as the zebra calcaneus, which is the heel bone, is dated to be between 2.5 and 2.1 million years ago. And in fact, everything that I've mentioned so far is based on butchery evidence of larger animals. But this talk is about smaller animals. So how do small game fit into this narrative? Well, not to give too much away, but for the most part, we don't really know. So right now, the oldest evidence of butchered small game extends back to only about 1.76 million years ago from a site called Olduvai Gorge in Tanzania. So there's actually a big difference between the oldest known butchered large animals compared to smaller animals. And I would also argue that the record for small game is less well known compared to larger animals. And there are many factors that play into this, but the fact remains that we do know more about the role of larger animals in our diet than we do about smaller ones. And we also don't view or conceptualize hunting smaller animals the same way that we do larger ones. So just for example, imagine trying to capture this rabbit or a rat or any other kind of fast-moving creature that's small without the use of traps or snares or nets or anything that could help you collect a large abundance of these animals. So I bet you're thinking that that seems like an extremely tiring task, that you'd probably exceed more calories chasing the rabbit than you would when you actually eat it. And this has actually been the prevailing kind of framework for hunting small game, that it's energetically costly to hunt a small, fast prey without the use of technology like traps or snares, all of which are so far only made by, or known to be made by, modern humans. So even in the case of Neanderthals, there are arguments that Neanderthals were not capable of regularly hunting small, fast prey, even though there is a lot of evidence that shows that both Homo erectus, Neanderthals, and archaic modern humans were consuming small animals, and that small animals are still being viewed as being too difficult or costly to catch, so it's not worth their time. So one of the questions that I'm very interested in exploring is, how did our ancestors, millions of years ago, hunt and consume smaller animals? And potentially more important, what kinds of technology or even cognitive capabilities, so how big potentially does the brain need to be for hominins to rely on or hunt smaller animals as a dietary resource? So before we answer that question, I want to share with you some of my own research on the topic that comes from Indonesia, and to give you a little bit of an idea about the kinds of evidence we're using to better understand how small animal assemblages are made. So shown here is a map of Southeast Asia. And for those of you who may be less familiar with this region, the top of Australia is actually shown at the bottom of the screen, and Philippines are the many, many islands that are shown in the middle top portion of your screen that's labeled. And the beige outlines are the current coastlines. So this is what the region looks like today. But at around 20,000 years ago, there was a lot more glaciers around, and so sea levels were quite low, and which exposed a lot more land in the shallow regions. So the gray areas actually represent the coastlines at that time. And so because this is a very unique geological feature of this area, there are several what we call biogeographical barriers, known as the Wallace Line as well as Lydekkers Line. each of which kind of denotes the edge of the continental shelves. So one shelf is referred to as Sunda, which is to the west, and then Sahel, which is to the east. And then the region in between these two barriers is known as Wallachia. And so this region is known for containing islands that have never been connected to continental mainlands and is home to many different kinds of plants and animals that are not found anywhere else in the world, including a lot of smaller animals. And so we've also known that our human ancestors have inhabited the region since as early as the 1800s, when fossils known as Homo erectus were first found on the Indonesian island of Java, dating as far back as 1.8 million years ago, and until as recent as 100,000 years ago. And then the discovery of Homo floresiensis at Liang Bua, and then also Homo floresiensis-like remains. These are remains that look very similar to Homo floresiensis from a site called Menenmenge on the Indonesian island of Flores. And this was the first skeletal evidence that any ancient human or relative crossed the Wallace line into Wallachia. And then more recently, a new hominin was described in the Philippines and named Homo luzonensis. And then there are, of course, other notable sites with cultural artifacts associated with ancient humans in the Philippines, as well as in the Indonesian island of Sulawesi called Telepu, where stone artifacts are dated to around 118,000 years ago. But the site that I would like to talk about today is located on this island here called Flores. And so this site is called, it's absolutely an incredible archaeological site, and it's called Liang Bua, which is a beautiful cave, as you can see here, where a human relative was unearthed and named Homo floresiensis back in 2003. And so the discovery of this partial skeleton, which is shown on the far right of your screen, caused a lot of controversy because the remains looked like something that should have been found in Eastern Africa at around 2.5 million years ago. But this the skeleton was unearthed in Indonesia and dated to only about 60 to 100,000 years ago. So very much out of place at a time. So her height, based on a couple of dimensions, is estimated to be about just over 1 meter tall. So this is about the height of a 5-year-old kid, and then had a brain size slightly larger than a chimpanzee. So since her discovery in 2003, scholars have proposed several explanations for where Homo floresiensis came from. The first is that Homo floresiensis represents a dwarfed population descended from Asian Homo erectus, so the Homo erectus material that's been found on the neighboring island of Java. The other hypothesis is that Homo floresiensis represents a population descended from a small-brained and small-bodied species of Homo. We're not sure which species in particular or what population, but that a lot of her anatomy resembles those of early Homo species like Homo habilis or rudolfensis that's found in Eastern Africa. And then the other hypothesis has been proposed that the skeleton is either a pathological or pygmy population of modern human, but this idea has more or less been rejected from the scientific community. What I find really interesting are actually the animals that are found in association with Homo floresiensis. So first, there's actually a very short statured elephant relative known as Stegodon florensis insularis, which stood at about 5.5 feet tall at shoulder height. So quite short little elephant relative. And then we have a giant marabou stork that stood at 6 feet tall. And yes, that's not a typo. This marabou stork stood at around 6 feet tall at Liang Bua. And then there's a species of an African vulture that was found, and also Komodo dragons, which are still found to be inhabiting nearby islands today. And there are also a lot of rats, a lot of rodents. And some of these were actually the size of a large house cat. And so the numbers I'm showing here on the screen next to each of these animals is actually the percentage of identified bones of that animal within the assemblage. And so if you look at the giant rats, 87% of the bones recovered during the time of Homo floresiensis were actually rat bones. That's a very large assemblage. We're talking about 300,000 elements identified as rats. So other than the fact that these rats are the most abundant animal at Liang Bua, why would we want to look at them? Well, there are a lot of rats in this region, and to just kind of visually demonstrate how many, here's what we call a circular phylogeny of rats that are found in Southeast Asia. And so all you need to take away from this image is that there are a lot of rats living in this region. And so in addition to the many species, rats also occupy a wide range of habitats. And so they can tell us a lot about what the environment was like back when Homo floresiensis was living in the cave. And so Flores actually has several rats that are endemic, meaning they're not found anywhere else in the world, not even on neighboring islands. And so they range in body size from something quite small up to, again, something like the size of a house cat. And if you've never seen rodent teeth before, now you have. So shown on your screen are rodent tooth rows of our endemic Flores rats. And so you can see just how different they are in size. And interestingly, we know that they prefer different kinds of habitats. So the idea here being that if you find more of one species of rat, then that might tell you something about the kinds of habitats that are nearby. And so my job as a zooarchaeologist is to figure out how these giant rats got there. How did they accumulate over time? And in many cases, a large bone assemblage like this is primarily due to predatory behavior. And so Flores is actually quite unique in that there are no endemic mammalian predators like foxes or coyotes that are also known to kind of hunt and accumulate smaller animals and create these bone assemblages. So at Liang Bua, the only predators capable of creating such a large assemblage of smaller animal bones are going to pretty much be limited to owls or other birds of prey or humans. And so let's first look to see what kinds of evidence owls leave behind. So when owls consume their prey, they tend to swallow it whole and then regurgitate the undigested portions in the form of an owl pellet. And then these pellets and then the digested bones contained within them accumulate over time. So shown here are two rodent teeth with arrows pointing to damage on the enamel, so the outside of the tooth, caused by owl stomach acid from being digested. And so what I did was I looked at the hundreds of rodent teeth from Liang Bua and counted how many showed evidence of digestion and how many didn't. And then I measured the width of the tooth to essentially use this measurement as a proxy for body size. And then I graphed the results. And so shown here is the density or the approximate volume of rat teeth with evidence of owl digestion shown in the gray curve, and then teeth without evidence of digestion shown in the blue curve. And so the takeaway here is that during this time, between about 120 and 190,000 years ago, owls are primarily eating rats of small and medium body sizes, while something else, potentially another predator like humans, are consuming rats of similar body sizes. And so what does the evidence for human damage look like? Well, here on the left, this is a 3D model of a rat humerus, which is the long bone in the upper portion of your arm. And next to it are 3 types of images showing 2 different marks on the bone surface. So the first are photographs of the marks themselves. And then in the middle are profiles or cross sections from a micro CT scan showing the internal structure of the bone. And then on the far right are 2 images of the marks on a 3D model. And so while owl damage is actually quite diagnostic or easy to identify, relatively, damage caused by humans can be actually quite challenging because behavior never repeats itself. So no mark will ever match another mark. And so it's our job to figure out how these marks compare to butchery marks created from experiments. So what are similarities and what are some differences? And so one way we do this is to collect information about these marks using 3D imaging techniques. So I'm currently collaborating with scholars at Colorado State University to scan a bunch of marks from Liang Bua and collect data that captures both the size and shape of each mark. So here's just an example of this where we can measure both the depth of the marks as well as the volume, and then also collect basic linear dimensions like length and width. And so the idea here is that this information will be compared to marks of known origin and we'll be able to confirm whether the mark is from a stone tool or potentially from a human tooth mark from chewing on the bones or from another predator. And so this work is ongoing, so I'm going to just kind of quickly summarize what we know so far about human activity at Liang Bua using just rodent bones. So on the far left are two groups of humans at Liang Bua. So I had mentioned that Homo floresiensis was found at Liang Bua, but we also have evidence of modern humans in the sequence, but they're non-overlapping, so they're quite distinct from one another. And so on the bottom is Homo floresiensis that's dated from around 120 to 190,000 years ago. And then we have modern humans from around 18,000 years ago to the present. And then I just kind of signaled when our agriculture starts to develop, which is around 3,000 years ago. So I'm going to show you a series of data. So here are just the number of bones that have been exposed to or have evidence of burning from fire. And this is according to spits. So the y-axis here, all those numbers going down are what we refer to as spits, which are 10 centimeter excavated portions of sediment. So as we excavate down, we excavate in these 10 centimeter increments, which we refer to as spits, while also following the natural geological formation of the sediment. And so a good way to read this is that everything that lines up with the blue line is from a modern human layer. And then everything that lines up with the orange line are from a Homo floresiensis layer. So right away, you might notice that the rodent bones from the modern human layers are burnt, while not one bone associated with Homo floresiensis was found had any evidence of burning. So this leads us to infer that modern humans were using fire to potentially cook rats and other animals, but Homo floresiensis was not. And next, we have the frequency of bones with marks made by humans. So I don't want to get too specific about this, but the main takeaway here is that Homo floresiensis and modern humans are both eating rats. And lastly, we have the accumulation of different body-sized rats. So clearly what this is showing is that there's a large accumulation of what we are referring to as our medium-sized rat, which is shown in green, during the time of Homo floresiensis, while there's a greater diversity of rats accumulated during the time of modern humans. So it's again shown in equal distance to the blue line. And so based on our results so far, we can infer a range of human activity and how humans utilize the surrounding landscape just from looking at rat bones. And so to kind of answer the question, who accumulated the rats at Liang Bua? Well, it's clear that both owls and humans are responsible for accumulating an incredibly large assemblage of rat remains. And by looking a bit closer at the kinds of damage caused by owls, we can see patterns where owls are accumulating rats of different body sizes, but at different times throughout the sequence. So, which can tell us something about how the environment is actually changing over time. And then we also see that both Homo floresiensis and modern humans are targeting different body-sized rats. And we also see that they're choosing to process them differently. So for example, modern humans are using fire while Homo floresiensis is not. So that tells us a lot about the kinds of behavior, the tools, and the technology available to them. And it tells us what kinds of environments they were also exploiting. So just to kind of bring this back full picture, so what does all of this mean? Well, it turns out that our research at Liang Bua shows that a small brain in a small-bodied hominin is in fact very capable and able to capture small, fast-moving prey. So Homo floresiensis did not use complex netting or trapping technologies, not even fire, to collect and process these animals. Yet they were able to hunt rats that were both very large, again, the size of a typical house cat, and as well as some medium ones, so smaller ones that would have been theoretically a little bit more challenging. And even today, local communities continue to hunt rats, and they do so by exerting very little energy. They simply wait along a rat trail, and when one runs by, they collect it. And so it's a very easy and non-complex way to capture a fast-moving rodent. And so this research has led me to question a lot about our assumptions about how ancient humans hunted smaller animals, and whether or not they played a larger role in our evolutionary history than what has been previously known. But one benefit of conducting this research at Liang Bua is that the bones are so well preserved and there are a lot of them, so the signals are pretty clear. And so we're also able to look at and compare two different hominin groups. We have Homo floresiensis, which is a small-brained and a small-bodied hominin with very simple technologies, and they're able to capture smaller animals. But then also comparing that to modern humans, which have a lot more complex technology available to them. And it's very clear that in this context, smaller animals were very much a part of their diet, and neither the cognitive or technological differences between these two hominins hindered the capture of smaller prey. And so with that, I'd just like to thank my Indonesian colleagues and the Human Origins Program for hosting this talk. And also a big happy 20th anniversary to the discovery of Homo floresiensis, which was discovered 20 years ago last month. So thank you so much.
Awesome, Grace. That was fantastic. Thank you. We have some questions, but I might actually take the liberty of asking you the first question because it's something I've been thinking about.
Sure.
I appreciated how you sort of laid out that there's good evidence for butchery of large animals from earlier sites. Do you think that there isn't as good evidence for butchery of smaller animals because people haven't looked at the small animal fossils, they weren't collected as systematically, maybe they were processed in some other way? Like, why do you think there is, besides, you know, that hedgehog at Olduvai, why is there this discrepancy?
Yeah, so I think definitely what you had already mentioned is part of it. There's just a lot of differences in how small mammals have been perceived. So, you know, going back to even when archaeological sites were first kind of being excavated in all these regions, you know, people just kind of threw away the small stuff and kept the large things. So this is kind of a pattern that, you know, unfortunately started with the development of our discipline. And yeah, I think that, you know, larger bones are, I will say I've now worked with both, so I can say that larger bones are quite easy. They're a lot easier to work with. Smaller animals, it is very tedious work. It does hurt your eyes. You know, it's quite challenging to do it repeatedly, especially for such a large assemblage. So I think there are a couple biases involved here involving research, taphonomic, attritional. So there's actually a lot of evidence of small mammal exploitation in, say, like the upper Paleolithic, so with modern human assemblages. But the further you go back in time, it's kind of unclear whether or not it's a combination of just the fact that these assemblages, either they're not hunting them and therefore they're not available, or they're being susceptible to damage and they're just being erased from the record. And so I think it's a combination potentially of both, but it's really hard to tell. And so I think, you know, there's a big window for people who, if you're interested in this, you know, to get into looking at smaller animals from a zoological and a taphonomic perspective.
Awesome. Yes, it seems like there is a nice small mammal niche for people who are interested in this. I will go to a question from Niels. This is an interesting question. Are any nutrients more abundant in small game that would make them a necessary part of a balanced prehistoric diet?
So actually, there's a project that is working on this that I can't actually talk about. But from what my understanding is that, you know, they are actually very nutritious, and they have, you know, the same kind of nutrients that larger animals have. You know, you have a balanced, you know, carbohydrates, protein, and fat, as well as micronutrients as well. But, you know, when I was looking at rats in particular, I found that even just the domestic rat actually has a larger percentage of body fat than lamb does. And that's something that I talked about in my dissertation. And so I don't know if this is a universal for all small mammals. That's something that does need to get, needs to be researched. But I know for rats are actually very fat and does have the potential to contribute very, you know, a lot of those kind of fatty nutrients that is argued to have been a very important resource for humans throughout human evolution.
It's definitely interesting given from what I understand, for humans that eat meat, eating too much lean meat without fat is not something we can even process really well. So that's really interesting. Another question from Niels: how would you put small animals in the context of a diet that may have included birds and fish as other sources of protein?
Yeah, the birds and fish and reptiles and things like that, they don't get a lot of like discussion, or people don't really talk about them as much in the record. Bats too, bats are also probably a really cool and important resource that, depending on where you are in the world, might have been pretty important. But unfortunately, a lot of those kinds of animals, again, are even more understudied than small mammals. And so we don't really have a lot of that information. And the ones that we do, and people actually do start to look at them, you do actually see that humans were, eating a lot of those other kinds of animals as well. It's just that signal is so low because, again, bird bones don't preserve nearly as well as mammal bones. And so you have this taphonomic bias and this preservation bias that's kind of working against you. So you can't really compare like a one-to-one with all of these. You have to kind of take into those considerations.
So here's an interesting question from Anne. Were small animals preferred over large animals, or were they chosen because large animals weren't available or too hard to catch?
So this is a pretty big debate, in human evolution, especially when you talk about what was the diet before we started hunting big game. Many scholars think that, we were omnivorous. Some think that we were purely vegetarian, so we didn't have any animal protein in our diet beforehand. And so, we don't really, I think it's still pretty open. There's been a lot of hypotheses thrown out. But I don't know because chimpanzees currently also hunt, smaller animals, but they do it more for like sport. They're not really relying on it as an integral part of their diet. And so it's unclear when we started to rely on potentially smaller animals in our evolution, because or at least within our line, because again, the record is really spotty, and we don't have the resolution, at least right now. And I hope, maybe sometime in the future, we'll have a lot more data that will support either, a consumption of smaller animals maybe pre or before humans started going after big game, or even maybe it was in tandem with big game. It's kind of an open question at this point.
Yeah, that makes sense. Here's a question from David, who asked, given a lack of evidence of use of fire by Homo floresiensis, so presumably rat meat was not cooked but eaten raw, would that have brought any risk of disease, assuming rats as they are now were disease carriers?
So probably not. So the rats that we're talking about on Flores, they're just like any other kind of wild animal. And I think this is something that is perceived for like, domestic rats, or, we talk about the rats like being responsible or partially responsible for the plague and things like that, like they're carriers of diseases. And that's something that is very much within the Western world, I think. And, we kind of have, even you look at any like children's cartoons, rats are always the bad guys, right? And so we don't have a very good perception of, rodents or rats in particular, right? And, but in this case, these rats are incredibly healthy. They're, eating natural like fruits and leaves and insects and other kinds of, material that's just provided for them, like in the forest. It's just there, and they're very healthy animals. So no, I don't think that the risk for disease was, any higher than any other, natural carnivore prey relationship.
Okay, cool. That's helpful. Because we got another question that was asking basically the same thing. So here's a question from, it looks like Gowley. How would they cook the rats? Would they just roast them, or would they use any other methods?
So I think for at least the modern humans, they would essentially roast them over a fire, or sometimes they would kind of chop them up and then, eat them or cook them separately after they've been dismembered or something like that. So you had mentioned that I work with, or I've done a lot of ethnoarchaeology on the island as well. And so we do have a paper that was actually recently accepted that kind of goes into that a little bit with our modern community partners. So people who are currently living around Liang Bua who do, hunt giant rats today, and it's part of their diet. And what they do is, they'll roast it just over the fire to kind of pre-cook, if you will, the meat. And then that's actually easier to then chop up and dismember versus if you're dismembering, it raw. And so then they do that, and then they kind of put it in a pot with spices and, pour over rice. And it's actually a very delicious meal.
So have you tried it?
I have, and it's really good.
Excellent. So we have a behind the scenes team question that I think is a great question. Is there any evidence of percussion marks on the rats to indicate that they were trying to get at marrow? Because we've been talking about fat and other kind of like non-meat, but animal-based resources.
So no, we don't have any in the sense of so percussion marks are essentially marks that you find on big mammal bones from, hard, like you use a stone tool and you try to essentially break open the a long bone cavity to access that fatty nutrients in the middle there. And with rats, no, they tend to either be twisted or, there's other mechanisms that or techniques that people will use to access the grease and the fat from the bones, especially cooking. But if you don't have cooking, if you don't have, access to fire, to get at those you'd probably have to twist or something. Now, I don't have any percussion marks per se, but that one actual mark that I showed, I'm still exploring what it could be. But if you noticed in the cross section, it almost penetrates through into that medullary cavity. And so that might have been evidence of them trying to get at the interior, and they gave up. So maybe it wasn't energetically beneficial, or maybe they were just breaking them and eating them that way. But we don't have a lot of broken long bones that would suggest that they were eating the medullary cavity or getting that fat from inside, because they do have quite a bit of fat on the outside. So it might not have been necessary.
Interesting. Here's a good question from Urate, who asks, sorry, did I miss this? What was Homo floresiensis's toolkit for hunting or processing food?
No, that's a good question. And I didn't really specify, but basically Homo floresiensis used very simple technology, stone tool technology. So what I mean by that is it looked very similar to the old one. So it had, your cores and your flakes, and they were taking nodules and making just essentially sharp edge tools. We didn't have anything like symmetry that we see in the Acheulean, where you have these beautiful teardrops, hand axes. There's nothing like that. So everything that we've found so far is quite simple. So just kind of core and flake technology.
Urate asks another question. Is there any indication in the cave of accumulation of animal pelts or hairs? Maybe this wouldn't be preserved?
No, the hair and pellets aren't preserved, but we do think, and I have kind of a running hypothesis here, is that a lot of these smaller animal bones were potentially preserved early on, or at least any kind of post-depositional, or after the bones and the pellets have been kind of covered, the pellets might have helped preserve those bones inside. But for the most part, no, that material actually gets eroded or dissolves away pretty early on.
Yeah, that makes sense.
Yeah.
I'm going to go to another behind the scenes question. So there's a big debate in human evolution about hunting and scavenging and different ways that animals were acquired. Is there, given that Homo floresiensis, seems to have processed and eaten rats, is there any way to tell that they were hunted as opposed to like collecting them when they were dropped from owls accumulating them, maybe targeting young rats, or doing things that maybe would have been more like scavenging?
We don't quite know that yet, because one way to figure out, and maybe Brian, you would be able to chime in on this, but to determine whether or not something was scavenged or hunted, a lot of the times, at least for big game, that has to deal with what elements you actually find in your assemblage, right? So you're looking for the fact that when you're scavenging something, you take the elements that have the most meat on them, and those are the elements that tend to preserve in your assemblage. But with smaller animals, we don't have that same kind of signal. So you're not going to get just part of the smaller animal and bring it back with you. You're going to bring the whole thing. So that same method to determine scavenging versus hunting doesn't really apply to smaller animals. And so to figure out if they're, sourcing potentially from, an owl or something like that, that would be really interesting. I'm not sure actually if a comparative, like a modern comparison, has actually ever been made. So we don't really know what that assemblage might look like in terms of which skeletal elements are preserved versus also what kind of damage would be there. Would you see a combo then on the same bones of owl damage and human exploitation? So I will leave that up for the next generation of taponymists to figure that out. But that sounds like a really cool project if anyone wants to do it.
Yeah, it's an interesting research question, I think. I think even with large animals, sometimes it's tricky to know exactly how an animal was acquired. We might be able to see what parts were processed, but, to know how an animal, like, was it killed by humans versus other predators, that can be really tricky, like you said. All right, I have another fun behind the scenes question. Since there's a possibility of an overwater crossing getting these hominins or maybe their ancestors to the island, is there any evidence of utilization of aquatic or shoreline resources?
So not so much at Liang Bua. So at least with Homo floresiensis. And then when modern humans come in, does seem to be some changes, or at least exchanges of, between freshwater and saltwater resources. But for the most part, everything does seem to be pretty mainland. So because the cave site is actually located pretty much center on the island in terms of north and south. And so it's not really close by, relatively speaking, to the coast. And so most of the resources that we are finding are just within like the internal region of the island, not so much the coastal.
Makes sense. Here's a good question from Urate again. Has anyone done an analysis of like tooth plaque looking for plants, other vegetable matter, anything like that?
Like on Homo floresiensis?
Yeah, I think so.
No, we haven't yet done anything like that. So that's we're definitely open to, conducting those additional levels. So, and we actually hint on the fact that, there are other ways to get at, understanding diet. And one way is to look at the surfaces of the animal bones, but another way is to look at, the remains essentially, right, of the plaque that's stuck in your teeth. And that will actually give you clues as to other kinds of foods that, Homo floresiensis was consuming. But no, that analysis has not yet been done.
Okay. Well, future research, it sounds like. That's cool. I have another question. Can you tell us what you're working on now as far as Homo floresiensis?
So now I've actually taken a break from the rats and I've switched over to the stegodon. And so right now I'm looking to see, how did Homo floresiensis collect the stegodon remains? Because this is a big question. Right when Homo floresiensis was discovered, it was immediately described as hunting, these elephants, these smaller bodied stegodon. And, the evidence for that was pretty premature, is very preliminary, but no one has kind of gone back to that since. And so what I'm doing is I'm looking at, a large portion of that assemblage, and I'm looking to see how did Homo floresiensis, acquire them? Were they hunting them or scavenging, or what's the evidence for hunting that has already kind of been proposed? So that's what I'm working on right now, but I'm also in the middle of getting a lot of this research published in the meantime. So
nice. Excellent.
Stay tuned.
Is there any thinking about that? Is there any chance that the stegodon, I mean, it sounds like some of the rat bones are in the deposits at Liang Bua, not just from Homo floresiensis or modern humans, but from owls and other birds of prey. Is there any chance that any of the stegodon might have like somehow gotten there naturally? It's hard for me to imagine that kind of scenario, but what do you think?
It is hard. So for at least the stegodon, Homo floresiensis could have accumulated them, but then also Komodo dragons, right? Komodo dragons is the apex predator on the island. And so they would have been going after stegodon for sure, because stegodon is the only large-bodied herbivore on the island. So that relationship would have been very strong. But the role that Homo floresiensis played as another predator would be really interesting. So yeah, we're not quite sure yet. In terms of them getting there naturally, I'm not really quite sure what scenario that would be. I mean, I know that at least African elephants, when they think they're about to die or something, they tend to leave the herd or die in isolation. So maybe they, went over and found Liang Bua is a wonderful place. I mean, it has this like natural air conditioning to it, and it attracts a lot of animals. So it wouldn't surprise me if, other animals on the island just, were naturally just attracted to it, and then they happen to, to die there. So it's a possibility, but again, I think all of this at this point is conjecture. We need to find the evidence to, whether or not, did Homo floresiensis hunt or scavenge them from Komodo dragon kills, and to what degree, are the stegodon, or how did they really accumulate? How did they get there? So that's the big question.
So following on, a question just came in from Walter who asked, could stegodons have been domesticated? And I have a sort of PS to that question. Are there stegodon in the layers with modern humans, or only Homo floresiensis?
Only Homo floresiensis. So no, there's no evidence of like domestication, not like what we see with like pigs and, other kinds of domesticated animals, but no, so they wouldn't be domesticated. But it is really interesting that they are only found, at least in situ, because there's a lot of erosional vents and things like that. So at least the material that we found in situ in those layers that are still kind of geologically intact, they are only found in association with Homo floresiensis. So we don't know what role like modern humans potentially played in their extinction. I mean, the same thing goes for Homo floresiensis. We don't really know yet what caused them to disappear from our records. Modern humans come in very, very soon after, so it's a potential, and that's kind of, the big question. Did modern humans, when they arrived on Flores, did they cause the extinction of both Homo floresiensis and then as well as the stegodon, the stork and vulture, all of them kind of also disappear? So, it's an interesting idea, but we don't have any overlapping evidence for that. And so we don't have any interaction. We don't know what could have happened. So there are a couple of hypotheses out there, including volcanism and other kind of environmental changes that happened also at that time that might have influenced, why we don't see them in the record.
So I see that this question is being answered behind the scenes, but I'm also going to pose it to you. Jay asks, what's the range of Homo floresiensis? And maybe you could tell us both like geologically, but also geographically.
Yes, so Homo floresiensis geographically has only been found on the island of Flores. And so it hasn't been found anywhere else. And temporally and through time, the oldest, I guess, archaeology that's been found has been dated to around 190,000 years ago. And then archaeologically, the soonest is about 50,000 years ago. So it's really within that with the archaeological material. So between 50,000 and 190,000 years ago. But then the skeletons of Homo floresiensis have been further kind of bracketed, and they're in this intermediate zone of about 60 to 100,000 years ago. So the archaeology kind of extends that a little bit. But yeah, and then modern humans come in at around like 46,000 years ago.
Or 4,600 years ago. Yeah.
46,000. Yeah.
Oh, 46,000. So there's a sort of
There's
4,000 year gap between. Okay.
yes, it's like
Yeah.
this. It's very, very
Wow. Okay. Here's a question about a different hominin species. This is a behind the scenes team question. Is there any indication that Homo luzonensis, and maybe you could tell us about that taxon and why I'm asking about it, maybe may have had the same sort of diet or toolkit as Homo floresiensis? Do you think there's any link between these two species?
So it is really interesting. They're different, or else, that's why it has its own species name. But being found on the Philippines is another island that again is kind of across that geographical barrier. So it is very interesting. But Homo luzonensis is dated again to about the same time frame as Homo floresiensis, roughly. But yeah, stone tool wise, very similar. And actually, there's a really cool rhino butchery site that is on the island of Luzon that is called Kalinga, which I mentioned, but it's dated to around 700,000 years ago, which is much older. And so, maybe something like a Homo luzonensis was potentially involved with, butchering up rhino on the Philippines, which is really interesting. So, potentially very similar. But there hasn't been a lot of research so far have been done with luzonensis. So it'd be interesting to see that comparison between the two hominins.
So it sounds like luzonensis is dated sort of later in time, like Homo floresiensis, and the rhino butchery is archaeological evidence that from the same place, but much earlier. Okay. Here's a question from Urate. How many Homo floresiensis individuals have been found?
So there is just that one partial skeleton, but there's also been a couple of other like fragmentary remains. And so based on which element and side, we can kind of narrow down, it's somewhere between 9 and 12 individuals.
Oh, wow.
So we actually have not yet done like a proper, even just zooarch analysis on Homo floresiensis to really nail down what is that, what we call the minimum number of individuals, so the MNI. But in the published literature, it's currently between about 9 and 12 individuals.
Okay, cool. Is there any evidence, Jay asks, of ritual burials by Homo floresiensis?
No, we don't have any evidence of burials by Homo floresiensis. So all of the material that has been found so far has been, pretty scattered, and there has been any kind of like symbolic orientation of the skeleton in any way. So, they're not like, arranged in a way that would suggest culturalistic kind of burials. And also just the formation of the sediment around when it was being excavated. There was nothing that resembled a burial pit of any way.
Okay. I'm looking to see if there's more questions coming in, more behind the scenes questions. I will ask another, what do you think is the most challenging part of the research that you've done so far, either on the rat bones, the stegodon bones, anything like that?
Oh my goodness. There's so many.
There's so many bones.
There's so many bones. Yes,
Yeah.
there's so many bones. So yeah, no, sorry, not so many challenges. But no, there are just so many bones. And with one of the challenges that I've noticed is that at least with the big animals, they're pretty fragmentary. So there's not a lot that you can really record from them. Right. But with the smaller animals, they are very much intact. We see all the skeletal features, and they're actually pretty clean in terms of visibility. You can see the surface really well. And so that, while great preservation, it also means that I can collect a lot more information per element. So it actually takes a lot more time, to go through each individual bone. And that's probably has been one of my biggest kind of challenges, is just that it's taking a long time. So the endurance for, going through and looking at this assemblage, it has, again, over 300,000 elements to it. That's a lot. And to be able to secure a pretty hefty sample, of that is taking quite a long time. So
Yeah.
And I want to do it right, and I want to do it, thoroughly. And so, we're kind of making sure that that is done right.
Yeah, I mean, 300,000 definitely sounds like a marathon as opposed to a sprint. So,
It is
um,
definitely a
I,
lifetime or more. Like, not going to finish that
exactly,
in my lifetime.
exactly. So we have a couple of more questions and then we'll wrap up. One is from Christoph, who asks, is there any evidence for Homo floresiensis outside of Liang Bua?
Not yet. So
Okay.
there are some hominin fossils. I mentioned a site called Metamenge, which
Mm-hmm.
is over on the eastern part of the island. And
Mm-hmm.
it's dated to, much earlier, to around that 700,000 time period.
Mm-hmm.
And they look very similar to Homo floresiensis, but so far they've never been assigned, specifically to that taxa. But they've been, or taxon, but they've been described as being, quite similar. They're just pretty fragmentary. And so, I think it definitely is related to probably some form of a Homo floresiensis population. It is much older. So
Mm-hmm.
we kind of have this like very old record and then this recent record, and we don't really have a lot in between. And so that's where a lot of our field work is also trying to be, concentrated, is in finding those sites that do give you that window, like in between, from what we know at Liang Bua and then what we know at these earlier sites that go back to like, a million years ago.
Mm-hmm. Good luck. I hope you find some good things in
Thank you.
between. Last audience question, and then I have one last behind the scenes question. So Jay asks, has there been any DNA testing of them? I think he means Homo floresiensis. And do they have the FOXP2 gene, the ability to vocalize?
So we don't yet have ancient DNA from Homo floresiensis. So ancient DNA doesn't preserve too well in warm and wet kind of climates. It really degrades the proteins. So, we have not had any luck so far in getting ancient DNA from Homo floresiensis. So, I mean, that is something that we're obviously very interested in getting. And, but that is going to be something that has to probably happen in the future with more new methods and things like that, because right now, no, there's not a lot being preserved at the cave. So, like, for example, I think collagen is only preserved as far back as like 3,000 years ago at the cave. So, not so much.
And actually, two more questions came in that I think are great. So I'm going to ask both of them before we finish. One is from Jay, who asks, is the size of Homo floresiensis related to their geographic isolation? As in, are they small because they evolved to be smaller?
That is a huge debate. In trying to understand, where did Homo floresiensis come from? So, so I didn't really touch a lot on that, but I did hint to it. So, so one hypothesis is that Homo floresiensis is, a relation or a close relative to, Homo erectus, because we know that Homo erectus has been in the region, like I said, since the 1800s. We've known about that. And so the idea is that a population of Homo erectus from the neighboring island of Java, they made its way over to Flores and became isolated and essentially just evolved in situ in that environment. And so one evolutionary process that's unique to islands is that animals that are naturally pretty big on continental mainlands become smaller, and then animals that tend to be smaller on continental mainlands get bigger. And this it's a complex system, but basically it has to do with an ecological release from predators and other kinds of, different kind of nutrients and things like that. And has to do with like food. And it's complex, but basically, yes, things get smaller or things get bigger. And so that's one explanation for why we do see this very short hominin. But the other hypothesis is that, based on her skeleton, or Homo floresiensis and the main type partial skeleton, the bones actually resemble pretty nicely, a late Australopithecus or like an early Homo. So specifically the pelvis and also the arm to leg proportions are similar to what you would expect at that time period. And so what we know about Homo erectus postcranially actually only comes from Africa. We don't really have any secure, confident postcrania of Homo erectus in Southeast Asia. And so that complicates things too. So what does the postcrania look like for an Asian or a Southeast Asian Homo erectus? We don't really know. You know, we can kind of hypothesize, but we don't really know what that looks like. And so when we talk about what does the whole skeleton, right, not just the head or the cranium and the teeth, but the whole package, what does that look like? You know, it does to me look like something that's very close to an early Homo, not so much a Homo erectus, but you know, I could go down a rabbit hole with this. So I'll kind of leave it there and, you know, point you to the literature, you know, to look at some of the other ideas and things. So.
Yeah. So we don't really know. I think it's a very good answer to that question. Yeah. Last one before we wrap up. Selquist asks, who funds your research?
So I have been funded by National Geographic. Actually, the Smithsonian Human Origins Program has also funded a lot of this research. Also the National Science Foundation and Winter Gren, which is an anthropology specific foundation. Also by the PEO Scholar Awards. This is like a Women in Science kind of scholarship. So those are just kind of a few of my bigger grants that have funded a lot of the research related to the rats, the ethnoarchaeology, and some of the more exciting stuff that's coming soon. So
excellent. Well, thank you so much, Grace. So we'll wrap up today's virtual program. Please also, please join me in thanking Grace for sharing her work with us. I'd also like to give special thanks to those who made this program possible, including our behind the scenes team who helped sort through and answer some of your questions, particularly Ryan McRae from the Smithsonian's Human Origins Program, to our donors, volunteers, and viewers like you, and finally to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. Our Hot Topic program next month will be on November 16th at 11:30 a.m. Eastern Time, and it is Your Inner Neanderthal, a deep dive into the Neanderthal and Denisovan DNA in modern human genomes with UC Berkeley geneticist Lauritz Skov. We've put a link in the Q&A where you can find information about our upcoming programs and how to sign up for the museum's weekly e-newsletter. That's really the best way to stay informed on upcoming programs and learn more about the museum's research and exhibitions. After this webinar ends in just a moment, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to Grace, to the participants, and to you, the audience. We'll see you next month.